Hello, everyone. This is Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium. I want to welcome you to our webinar series today. We are joined today for today's webinar by Gabriel Keeble-Gagnère from Agriculture Victoria. But before we move to Gabriel's talk, I'm going to give you a little bit of an overview of the International Wheat Genome Sequencing Consortium. So we have more than 3,200 members in 71 countries and almost 900 institutes, research institutes and companies involved in the IWGSC. And we have 8 sponsors who make our webinar and our activities possible. So I want to thank all of our sponsors today. And our newest sponsor is Illumina, who is the subject of today's webinar. So our vision is for what we call 2.0, which is post the reference sequence that we released in 2018 and published in 2018, is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. So we have a number of different activities. We're continuing our Arbor Biosciences collaboration. We expect to make an announcement shortly regarding the open access and release of RefSec version 2.1, as well as RefSec annotation version 2.1. We continue to try to secure funding for 8 land races that would represent the breadth of diversity in wheat. And we also work to try to get others to release genome sequences for elite varieties and other genomic resources. Our next webinar, which will be in May, is how a wheat disease resistance gene atlas could lift up wheat breeding, and it will be done by Amber Hafiz from the John Innes Center, and we encourage you to register for that and participate in that webinar as well. Just so everyone will know, this webinar is recorded and will be posted on the IWGSC YouTube channel. You can subscribe to the channel so that you never miss a new upload. The presentation will be followed by a Q&A period. Please submit your questions in the Q&A panel and use the chat only to talk with fellow attendees or the organizers. Already, you can download the presentations in the handouts link, so feel free to do that. So without further ado, I'm going to turn it over to Gabriel, who will give us a talk about the novel design of imputation-enabled SNP arrays for dual hybridization of wheat and barley. Thank you, Gabriel. I'll turn it over to you.
Okay. Can we all see that the first slide there?
Yes, we're good. Great.
Okay, great. Okay, so let me give you one moment. Okay. All right. So thanks to Kelly for that introduction. Thanks to IWGSC and Illumina for inviting me to talk today. Thanks everyone for tuning in. And I'd like to thank all of my colleagues from Agriculture Victoria and Intergrain who have been part of this work, and particularly those who have helped me put together these slides for the talk. Okay, firstly, I just need to make it clear that the views expressed in this presentation are my own and do not represent the opinions of Illumina. And I just want everyone to just have a moment to read that themselves. Okay, and I just want to start by introducing the organisations involved in this work. The first being the organisation that I work for, Agriculture Victoria Research. We're part of the state government of Victoria in Australia. We are based in Bundoora with sites around the state. And we're tasked with supporting agriculture through research, innovation and building strong science capability. The second organisation is Intergrain, an Australian breeding company focused on wheat, barley and oats. They run national breeding programs around Australia with more than 300,000 plots all over the country. They're a highly innovative company, as demonstrated by their desire to work with us to bring genomics into their programs. They're also very open to collaboration. They were one of the key drivers behind making the chip publicly accessible, as they saw it as a way to give back to the community and also to drive collaboration, ensuring that we can meet the challenges facing global food security together. So there have been major advances in wheat and barley genomics in recent years. I think that's not news to many people listening now. So we now not only have high quality reference assemblies in both species, which continue to be improved, but also multiple assemblies of comparable quality of elite varieties in both species. In addition, there are now extensive genome scale genomic resources exploring diversity, and all this is really being driven by the ability to genotype, which we can see reflected in the range of options now available. But then this range of options presents a challenge when we're considering what the best choice is for our particular application. When we consider the needs of researchers and breeders, we see that they're quite diverged. And for us, thinking about our role supporting pre-breeding research, but also large-scale breeding applications such as this collaboration with Intergrain, we didn't want to use separate platforms, but rather wanted a single platform to meet all the needs that we were going to face. However, we felt this wasn't available. So we concluded that we needed to design a new genotyping platform, in particular, an efficient genotyping platform that could provide this bridge between research and breeding. And in particular, this is nice not only because it means we only have one platform to focus on, but it enables information from research to flow through to breeding and vice versa. And we chose Illumina's Infinium XT technology because it was the technology best suited to our needs. In particular, we knew we needed to be able to deploy it at large scale. By that I mean hundreds of thousands of samples per year. XT technology has a demonstrated track record of enabling this. So why didn't we just use an existing technology? We're aware that using subsets, for example, of the wheat 90K array is an approach often used, but the 90K was designed before these advances in genomic resources that I described at the start. We felt that we could leverage the newly available resources to design a more informative and cost-effective chip. And importantly, really it was the cost element that we thought that we could really drive down and create a much more cost-effective chip. So there's plenty of information online from Illumina about how the chemistry behind XT technology works. One thing worth noting for those not familiar with bead chip-based technologies is that it works by detecting fluorescences from dyes attached to the probes on the chip, and the relative intensities determine allele state. So what this means in practice is that probe design is going to be important to ensure good, clean data. And of course, in practice, large-scale deployment of genotyping chips means there's the inevitable day when they arrive, together with large quantities of dry ice, and it's great to see senior management involved in unpacking the boxes here. So considering our aim of bridging research and breeding applications, we came up with a few objectives that we wanted to keep in mind throughout the design process. These revolved around careful probe design to ensure good quality data, as I just mentioned, ensuring we used maximally diverse germplasm to reduce ascertainment bias and enable the chip to be used in as many and as diverse situations as possible. Cost was a big factor, so we wanted to explore the potential for dual sample hybridization, and we wanted to include content to enable specific research and breeding activities. The challenge really was to balance these objectives while keeping SNP numbers as low as possible. And as I noted, probe design is critical to ensure clean data. In particular, especially important for polyploids like wheat, is that probes targeting multiple loci cause cluster shifts, which we see here. And this results in it being more difficult to call genotypes, particularly for hets. So to mitigate this, we wanted to design probes targeting unique loci wherever possible, and hopefully resulting in clean, separated clusters like we see here. Another way the signal can be disrupted is by variation detected in the probe sequence itself, which again causes cluster shifts and ultimately makes the SNP difficult to call. What this means is that we don't want to target SNPs which have another SNP a few bases before it, just before the target SNP. And otherwise, we'll end up with situations like this, and we see a lot of this in the 90K, where multiple signals are confounded and accurate calling requires further work. So we want to only consider probes that are free of variation in the probe sequence itself. And so the good news is that the availability of high quality reference genomes means that we can now informatically search for good probes and discard those that don't meet our criteria. And this is something we just couldn't do before. So that's probe design. I also said we wanted to maximize the diversity we're capturing to reduce ascertainment bias and make the chip applicable in as many situations as possible. So for wheat, we have an in-house collection of 7,000 or almost 7,000 globally diverse wheat accessions, from which we selected 1,200 accessions specifically to capture the genetic diversity of the bigger set. And we exome sequence those. This is a data set that we've used for many years. We're very comfortable with it, and we're very confident that this is a good sample of global wheat. In barley, we used an exome data set published by IBK, focusing on capturing geographic diversity of barley land races. We did do some work to establish that these accessions were a good sample of global diversity. We also had 120 historical breeding lines from intergrain, and in barley then, we were very confident that we had a good sample of global diversity too. So from these diverse accessions, we can call SNPs against their respective reference genomes. The good news is that we find many SNPs, so there's a rich resource we can mine for content to go on the chip. But now we have another problem because, as I said before, we're looking for low numbers of SNP to go on the chip to drive down costs. But here we have millions of SNPs, so we actually need to come up with some kind of process to select the best SNPs, if you like, to go on the chip. How can we do this? If all the SNPs were independent, we'd have a hard time trying to capture enough information with a small subset. But luckily, they're not. The term for this non-random association of alleles is linkage disequilibrium, or LD for short. We use the R-squared measure, which is a common measure of linkage disequilibrium. This is reasonable because it's very important in mapping applications and things like GWAS. This then gives us a way to relate SNPs and a relationship between the SNPs and, importantly, the information they carry. So an R-squared of 1 between SNPs means that they carry the same information, i.e. they are completely equivalent. An R-squared of 0 means the two SNPs are independent of each other. Now this R-squared measure is great because it means one SNP can actually give us information about others. when they're in high LD. So we don't need to consider all SNPs now. So as I said, an R² of 1 between SNPs means they carry identical information. For our purposes, we actually consider SNPs with R² greater than 0.9 to be equivalent. This is reasonable because we're looking at global diversity, and there can be exceptions in the relationship between SNPs, especially in exotic material. So we allow for a bit of variation there. We wouldn't get very far if we considered only SNPs with R² of 1. But this gives us a very nice equivalence measure and a way to group SNPs by what we call tag SNP set. So in this example, we have 6 SNPs, and I'm showing the R² between them here. And those that are circled in red have R² all greater than 0.9 between them. So we consider this a tag SNP set, i.e. we consider all these SNPs equivalent. So we don't need to consider selecting all of them. One of them will be enough, as it carries the information from the others. And the nice thing about this is, as I've said, probe design is important. We might filter out probes because they don't match the criteria that we need, for example, uniquely mapping. But because we've got a set of SNPs here carrying the same information, we only need one of them to pass our filters, and we can select it and capture all the information from that tag SNP set. Now, the size of the tag SNP set is a very important measure because it gives us a way to rank or to prioritize some SNPs over others. This ranking then leads us naturally to a new selection algorithm that we developed specifically for this task of SNP selection. In brief, the way it works is we calculate all the tag SNP sets at R² greater than 0.9. We filter the list based on our design criteria, and then we rank them in order of size of tag SNP set. And we just take the top SNP, i.e. the most informative SNP, the SNP with the most SNPs in its tag SNP set, put that on the chip. Then we remove all the SNPs in the tag SNP set and all the SNPs in each of their tag SNP sets from the list. Now we do this to avoid adding too much redundancy. At this stage, we don't want to do that. And we continue down until the largest remaining tag SNP set is less than a given number. And we actually run this down to 10, 20, and 50. The reason for that is that that ensures that the largest tag SNP sets are tagged multiple times. And this is what we want, because there's still the chance, for whatever reason, that a SNP fail. For example, there can be factors that we couldn't foresee that makes it hard to actually call from the clusters, or it may not even synthesize onto the chip. So in that case, we would lose the information from all those SNPs that it's equivalent to, whereas here we're mitigating that chance by adding some redundancy. And so this process then minimizes SNP number to maximize the LD capture. So it's useful to view what the algorithm is doing visually to get an idea of what's really going on and why this approach works. So what we see here is the number of tagged SNPs on the y-axis as we progress through the algorithm here each iteration. The first thing we note is that the curves for wheat and barley are very similar in that they start steep and then flatten out. What this means is that most of the informative SNPs we capture early on, and then eventually resulting in SNPs that are only really informing about themselves, i.e. they're singleton SNPs. So with very few SNPs, we're able to capture a lot of the information content in the SNPs. So for example, in wheat, with only 11,000 SNPs, we're actually tagging at R² greater than 0.5, almost 700,000 SNPs. And in barley, with 7,000 roughly, we're able to tag around 400,000. Now these numbers, say 11,000 and 7,000 SNPs to go on the chip, these are great numbers for a low density array. And these numbers that we're tagging, i.e. in the hundreds of thousands, are actually a good chunk of the SNPs that we started with. So we've found a way to capture a lot of information with very few SNPs. It's worth noting that this distribution is not uniform for every chromosome. As expected, the number of SNPs required to capture diversity on the D-genome, for example, is low, and we expect that. But the important point is that the algorithm principle is very simple and is actually naturally saturating areas where diversity is high and then selecting fewer SNPs where diversity is low. Like, we don't need to specifically guide it. It's actually driven by the LD patterns inherent in the data. Again, highlighting the importance of capturing that maximal diversity in the data sets that we started with. So until now, I've not actually mentioned minor allele frequency or MAF because the algorithm is MAF agnostic and we don't specifically guide it towards high or low MAF. Again, we just let the algorithm decide this. But we do need to consider MAF given the range of applications that we're targeting. In particular, in a biparental mapping population, any polymorphism between parents will be useful regardless of the MAF in a global context. But for applications involving global diversity, such as GWAS, we need rare variants to be able to differentiate divergent accessions and particularly potentially pick up that causative variant. So we need to balance high and low MAF, but the question is, how do we do this? So we actually think that the best MAF distribution is one that mirrors the original distribution that we started with. And that's in fact what we see. What we see here on the left is the MAF distribution for the exome SNPs that we started with in wheat and barley. Then we see the MAF distribution of the selected SNPs and the MAF distribution of the SNPs that are in R² greater than 0.7 with those selected SNPs. And we see that in all cases, we see the skew towards low MAF, which is exactly what we want because we know that good sampling of low MAF is important for diversity applications. Of course, because there are more low MAF SNPs, i.e. there are more rare variants in total, we need to include more on the chip. So it makes sense. So we've devised a way to select a small set of SNPs to go on the chip which maximize LD capture. This is great because we need to keep chip content low to reduce costs. But inevitably, with fewer SNPs, we have reduced statistical power relative to high density genotyping such as exome sequencing. But now we can use a statistical method called imputation to recover a proportion of the SNPs that we do not genotype and recover some of that lost statistical power. So just to be clear, for imputation, we have a reference population genotyped at high density. For example, the exome SNP datasets that we start with. And then a target population genotyped at a lower density subset of the higher density set. So for example, the SNPs that we've selected. An imputation then uses the haplotype information in the reference samples to infer haplotypes in the target population. We hypothesize that since we have maximized LD capture with our selected set of SNPs, we should be able to impute to these numbers that we're tagging at the various R² cutoffs with high accuracy. But of course, we need to validate this and actually test that it really works. So we define the target set of SNPs to be the sets of SNPs, as I said, into R² cutoffs of 0.5, 0.7, and 0.9. And we want to ask the question, how well can we recover these sets of SNPs, given that we start only with the SNPs on the chip in each case? Okay, and we use 100-fold in silico cross-validation to validate this test. We start with the exome genotypes. We extract, after filling in missing data, we extract a subset of 100 samples. And of course, these 100 samples are then not used as part of the reference set when we impute them. We impute with the rest of the samples. So this gives us an accurate measure of how well we can impute into arbitrary germplasm. And then we impute up to the various sets that we've just defined. So actually, we use we measure accuracy in two ways. One is the concordance, which is just straight proportion of correctly called genotypes. The other is correlation. By that I mean the Pearson correlation squared, R² between imputed and exome genotypes. The reason I use both these measures is that concordance can be misleading, and due to the bias towards low MAF, we can actually bias the concordance to high values, which I mean, may not really represent how well we're doing. So we use correlation, which is more sensitive and gives us a better idea of how well we're actually capturing the information carried by these genotypes. So here I've summarized in order of with the samples ordered by how well they're being imputed, and we can see actually overall the results absolutely validate our thinking in that in all cases, even in the largest set and with the strictest measure of accuracy, we're well above 90%, which is incredible. And it's only really a few samples at the end where we seem to see a drop-off, and this may in fact be related to problems with the exome data. So overall, we're very happy we can recover these genotypes with high accuracy. In barley, the story is very similar. In short, we're able to recover the genotypes at the various R-squared cutoffs at very high accuracy. We only see a little bit more of a drop at the bigger set, but remember, barley had fewer samples in the references, so this is perhaps not unexpected. But overall, absolutely validated our thinking and that we are able to recover these genotypes with high accuracy. So in summary then, we started with high density SNP. Through LD analysis and a selection algorithm that I described, we're able to determine SNPs to go on the low density Infinium wheat-barley 40K array. And then through imputation using the diverse reference haplotypes, we can actually recover, again, high density SNP with high accuracy. So in addition to being able to impute to high density, we can actually also use imputation to integrate different genotyping platforms. So if we consider genotype platforms commonly used today in wheat and barley, they typically assay tens of thousands of SNPs, but often the SNP discovery is done in a different way with different germplasm and different SNP selection process, the end result being that overlap of the content can be very low, ranging from say 1,200 SNPs in this case with these two assays in barley, which is not bad, to just a few hundred in some cases, and particularly when we consider the overlap between multiple platforms, we see really quite low numbers, in this case 99. This is really quite hard to work with. And so this makes it hard to integrate these integrate samples genotyped on the different platforms. And by integrate, I mean combine and use together in a statistical analysis. So the situation is transformed when we use imputation. Here I'm focusing on wheat, but the idea is the same in barley. So the idea is that we apply the same process I've just described for imputing the SNPs from the wheat-barley XT chip. So we use the overlap to the high-density exome data and impute to the set of SNPs in R² greater than 0.7, which we know can be done with high accuracy. Then when we consider the overlap of these imputed data sets, we're actually seeing, rather than 99 SNPs, now over 120,000 SNPs in common imputed with high accuracy. So this is exciting because it means that legacy data, and we may have lots of samples genotyped in the past with older technologies, using imputation, we can continue to use them, integrate them with samples genotyped on the XT. So this means we can reuse and extract more value from legacy data. Finally, I'll just note that the numbers that are being imputed to by the different platforms, we obviously note that the XT, by the wheat-barley 40K XT, stands out being particularly high. This is because it's been specifically designed for this task. So this goes back to the question I raised at the start, why didn't we use an existing genotyping platform? Well, none of them would be able to impute to these kind of numbers with such high accuracy. And this summarizes really what we're doing now. This is actually what we're doing, implementing genomic selection in integrated breeding program. A proportion of the training population is actually genotyped on the TGBS, which we, through imputation, we combine with the wheat-barley 40K. And it works very well. But the XT chip is actually designed for the future as well, of course. As we move to whole genome sequencing data in wheat, This will increase the density of SNP information that we have, which will naturally increase the number of SNPs that the XT is tagging, because obviously now with exome data, we have patchy data based on the capture space. But as we get whole genome data, we'll fill in these patches and be tagging more SNPs, of course. The higher density SNP will also enable further integration with legacy datasets, so we'll be able to do even better than I've just described on the previous slide. And further importantly, the process I've described for selecting SNPs actually gives us a way to continually update the chip content. So as our knowledge improves, we actually have a way for the Infinium 40K chip to evolve in parallel with that knowledge. So that summarizes the process for selecting the core of the content, which is around capturing diversity and enabling highly accurate imputation. We also wanted to add content to connect to existing knowledge, be it markers used by breeders or GWAS signals published in the literature. And also, given the richness of this IPK dataset, where they've GBS genotyped their entire gene bank, we wanted to ensure connectivity to this dataset, so we added a few hundred SNPs to do that. They may be thinking, well, why don't you just use the imputation methods that you described? But actually, when datasets are very sparse, it's actually a good idea to have a direct connection, and that can actually enable better results through the imputation methods I described. Then, as I said, we wanted to include content to enable particular research and breeding activities, considering the importance of synthetic wheats in introducing novel variation into wheat. We wanted to target variation specific to synthetics. So synthetics are the combination of a tetraploid wheat, the A and B genomes, and Aegilops tauschii, the D-genome progenitor of wheat. So we included a set of 2,600 SNPs directly from the 90K chip that we knew is able to differentiate between different groups of tetraploid wheat. So note here that I'm showing a PCA with all the 90K SNPs on the left here versus a PCA with just the 2,600 SNPs on the XT. And we can see that population structure is nearly the same. So we know that these SNPs are going to do a good job of differentiating the different subpopulations in tetraploid wheat, and that samples genotyped with the XT will be able to do that. It's also worth noting that the majority of SNPs on the chip are actually polymorphic in tetraploid wheat. So while it wasn't designed with that in mind, it can still be used with tetraploid wheat. For the D-genome part, i.e. Aegilops tauschii diversity, we selected SNPs enriched in the D-genome of primary synthetics. And we can show that this enables the XT chip to separate synthetic wheats out from standard wheats as well as the 90K chip does. So what these tetraploid and D-genome SNPs enable is both the identification of already integrated material, but also enables integration of new haplotypes and the ability to then track those haplotypes through a breeding program, for example. And that sums up the design for version 1. As I said, the bulk of the content is around tagging SNPs for imputation. And with extra SNPs added to connect to existing knowledge and resources and to enable specific applications. So in total, we have just under 40,000 SNPs. But what we really want to know is how well it performs. We can see here, Joss just couldn't wait to get the data, so he started calling genotypes by hand. Which I don't recommend. But so now, this goes back to what I said about at the start about the importance of probe design. What we can see here is that we've actually done a very good job. The majority of targets are single locus in wheat and barley, with some multi-locus in wheat, though this is expected because it's a polyploid. So overall, this is great news. And it translates to very good genotype calling, in particular low missing data rates, typically under 10%, and in particular very high concordance between the XT genotypes and true genotypes by comparing back to the exome sequencing of samples that we've run on both platforms. We could see really well above 95% in most of these metrics, and only a slight reduction when considering hets and the correlation in wheat, and I'd be willing to bet that actually the XT is doing a better job than the exome in this case. The source of the error here probably is the exome sequencing. So that's the performance of single hybridized samples. And that is, we start with a wheat DNA, we run it on an assay and we get wheat genotypes, or we start with barley DNA, put it on an assay and get barley genotypes. Now this requires 2 different assays. But what we're really excited about is the case where we can dual hybridize samples. That is, we combine wheat DNA with barley DNA, we produce this 28 chromosome synthetic genome, if you like, and we run that single sample on one assay. And then through clever training of the clustering algorithm, we're able to call wheat genotypes and barley genotypes. And of course, this uses 1 assay rather than 2, and so if it works, it halves cost. And note that I'll use the term single hybs and dual hybs in what follows, just out of habit. So it's worth noting that we didn't know if this would be possible initially. We believed that it would be, but obviously you don't know until you've actually tried it. So we've actually done it, and the results are incredibly good. We're able to call good numbers of SNP in both wheat and barley. It's also worth noting that we used very strict cutoffs, so in hindsight we could probably improve these results, but actually on the face of it, we're very, very happy with this. Particularly when we look at the quality of the genotypes that we're calling, we're essentially on par with the single hybs with comparable missing data rates and almost identical genotypes that we get back when we compare back to samples that we've run both as single and dual hyb. Now we do see some SNP attrition in the dual hybridization assays, but the important point to note is that when we consider how these are going to be applied, i.e. usually in large-scale deployment in breeding programs with material that is related, we are able to recover the SNPs lost to attrition through imputation using the single hyb samples as references. And finally, we expect to improve on these results over time. So we succeeded in enabling dual hybridization. So the chip is delivering high quality genotypes, that's good news, but we want these SNPs to be detecting meaningful biological signals too. To validate that this is the case, we can run a GWAS, and it's nice to do it on a trait where we know the QTL involved to validate that the chip is working. So in barley, row type is an easily observable and measurable trait, making it ideal for GWAS. When we do the analysis with XT genotypes, we see a number of peaks, 2 of which correspond to VRS1 and INT-C, which are known genes involved in row type determination. And we get a number of other peaks, which would be worth following up. So in barley, this is nice validation that we're actually picking up good biological signals. In wheat, we've done work on the AWN trait, again, easily observed and measured trait. We know it's controlled by 3 loci in the genome, 2 of which segregate in our population. 1 of them on 5A and 1 on 6B. Now, if we look at the exome data, as I said, we get this strong peak on 5A with a more diffuse signal on 6B. If we look at the XT raw genotypes, we actually detect the signal on 5A. We do also detect the signal on 6B, though similarly to exome where it's more diffuse than on 5A, it's obviously more diffuse than on 5A in XT as well. But importantly, once we impute the XT data, we see a much stronger peak in the 5A QTL. And this really demonstrates the value of imputation in recovering some of that statistical power that we lost. So in wheat as well, we're detecting real biological signals. Okay, so at the start of this project, we knew that large-scale deployment of genomic prediction in Intergrain's breeding program was one of the key use cases the chip was needed to enable. And it is being used in its 3rd year now, and it's working very well. I've summarized some of the results here with 3 traits in wheat and barley. We have yield, a disease trait, and a quality trait, each with quite different heritabilities. And these accuracies are very respectable and certainly capable of assisting breeding decisions to deliver genetic gain. So these are really quite different traits. So from the point of view of the chip, we're confident that it's providing us with the right kind of data needed to drive these predictions. But of course, the chip is not the only factor here. The size of the training population, the quality of the phenotypes is important. And as these data sets increase and improve, we believe these results will continue to improve with it. But already, we're very happy with this performance, and we are successfully implementing genomic prediction in Intergrain's breeding programs. So that summarizes the performance of the chip from a number of different perspectives. As I said, the overall performance has certainly surpassed my expectations. We're very happy with the chip and excited about other people using it. But of course, there's always room for improvement, and that's exactly what we've just done with some additions to the content in a version 1.1 update. This mainly addresses a few gaps in coverage, particularly on the D-genome of wheat to facilitate genetic mapping applications, and it adds more trait-associated SNP. And this illustrates this idea of continually updating the content as our knowledge improves. And this design process that I've outlined for wheat and barley is completely general. And by way of proof, we've already implemented it in a multi-species pulse chip targeting chickpea, lentil, field pea, and lupin. So it's the exact same ideas. It's already being deployed successfully, and we're actually even able to carry out quad hybridizations in this case. Now, going back to my initial point about the explosion of genomic data that we're seeing now, the question is how to process it and how to make sense of it, particularly if you don't have bioinformatics skills. And more to the point, now that we've designed this new chip, how do we facilitate uptake and integration with existing resources and knowledge? So to that end, we've developed and are developing a tool we call Pretzel, which is a web-based platform for interactive visualization and analysis of genomic and genetic data sets. It allows, for example, chromosome scale alignments between whole genomes, for example, using common genes or synthetic genes as the linkages. We can also align genetic maps to physical alignments and define QTL. And then we can go from the chromosome scale right down to base pair scale, interrogate genes and exome SNPs around the genes, and also ask questions about, for this given 90K SNP, what's the nearest 40K SNP, for example. And this helps relate results, for example, QTL discovered with the 40K to other QTL in the 90K. A classic question being, is this QTL the same as that QTL? And we can answer those questions with Pretzel with a few clicks. So in conclusion then, we've designed and tested a new wheat-barley 40K genotyping platform that bridges research and breeding. It delivers high quality genotypes in wheat and barley. It enables imputation to high density with high accuracy. It can dual hybridize wheat and barley samples, halving costs in high volume applications. And it's been effectively deployed in Intergrain's breeding program. Importantly, it's also being made available for use by the international community through Illumina. With that, I'd like to thank all the colleagues at Agriculture Victoria and Intergrain who have been involved in this project. Thank you, and I'm very keen to hear questions. Note that I'll answer questions around the design of the chip, but questions relating to Intergrain should go to Hannah Robinson, whose email address is here. And any queries about access to the chip should go to your Illumina representative. Thank you.
Great. Thank you very much, Gabriel, for a great presentation. As Gabriel mentioned, he's happy to talk or respond to any questions regarding the chip. If you have questions related to Intergrain, then those will be answered subsequently to the presentation. As most of you may know, we tend to post on our YouTube channel the Q&As that come in as well in writing so that they can be used for future individuals. So, and I'm not sure you can answer this one, Gabriel. The first question is, how much will it cost per genotype or assay, more or less? Do you know
Okay,
that?
Okay, that's something that's best to ask to Illumina. I'm sorry, I can't give an exact answer to that.
All right. So here is a question. Very interesting to see the concordance in results between single and double hybrids. Did you need to standardize the DNA concentration of the samples combined, or was there a degree of tolerance of different DNA concentration between the samples?
In short, yes, the concentration of DNA is important to get good results in the dual hybrids. That's something we spend a lot of time on.
Yeah. So, all right. So the next question is, if someone wants to study durum wheat land races for drought tolerance using SNP markers, which genotyping platform do you think is more reliable, particularly if you want to also incorporate GWAS?
I would say to try with the XT. I think that it's certainly most likely going to work very well. But at the same time, we don't see the XT as completely replacing all other genotyping platforms. It's solving a particular problem. There are going to be use cases where, for example, the 90K is still the best choice. But I would certainly try the XT on a subset of your samples, for example, and see how it performs. I believe it will work well in tetraploid wheat.
Okay. All right. So the next question is, you mentioned in your presentation that there were gaps in coverage in the D-genome. Do you have any idea why that's the case?
So, I mean, the D-genome is obviously low in diversity in hexaploid wheat, and we knew with the initial design of that SNP selection algorithm that I described, which is relying on LD, we actually knew already that we were having very few SNPs on the D-genome. But this made sense to us because at the time we were thinking about genomic prediction and capturing LD structures, and the data spoke for itself. But actually, in practice, because it's so sparse on the D-genome, genetic mapping in particular is more difficult on the D-genome specifically. So that's why we filled out those gaps. And in fact, it's not trivial to find good SNPs on the D-genome that passed all our filters, as suitable to be able to put on the chip. It's actually not that easy to do because
Yeah, continue, please. Yeah.
Oh, I was just surprised at actually how hard I had to go searching and finding SNPs to go on.
Yeah, so that which is part of the question is, how do you actually go, if you're not a bioinformatics specialist, how do you go about filling those gaps?
If you're not a bioinformatics specialist, I'm not sure. But really, this comes down to kind of augmenting data sets and particularly whole genome data sets. And in fact, that's what we had to use in the end to fill out all those gaps, was using some of the 15 varieties sequenced in Australia 10 years ago. And that was useful to find good quality SNPs.
All right, so the next question is, how is this chip better than GBS? And how did you make sure you limited ascertainment bias?
Okay, I'll just answer the second question, the second part first. I mean, we were very aware of the need to avoid ascertainment bias, and that's why we based the data sets on highly diverse germplasm, and also didn't bias the SNPs to high math, which is where you tend to get a problem with ascertainment bias. So as I showed, we have this skew towards low math, and this is really an important part in avoiding that ascertainment bias. Just for those who don't know, ascertainment bias is really when samples appear more similar than they are, and that's through too much high math. Everything starts to appear similar when in fact, because you're not capturing those rare variants, you're not seeing that they're as different as they are. And so then the first part of the question, why is it better than GBS? Well, I mean, with sequencing-based technologies, you have issues around, say, call rates and the quality of the data. And you may have to resequence further to get more data. And it's really about this. The thing with the XTs is designed to be deployed at very large scale. I mean, hundreds of thousands of samples, which is tricky with the GBS technology, I believe.
All right, the next question is, apart from manual scoring, which scoring algorithm did you use for allele discrimination?
Is that referring to the actual SNP calling process?
I believe so, yes.
Okay, so that SNP calling process is described in the Hay et al. Paper, published in 2019 in Nature Genetics. So this was a pipeline that we developed internally, and we're very happy with it because, as I said, like essentially all the SNPs on the chip return polymorphism. So it shows that our SNP calling algorithm is good, and really it just involves calling SNPs against the reference, and of course the IWGSC reference was integral in enabling the sort of exploration of that data to its full extent. Then, only calling SNPs when we see them in multiple samples, for example, just to ensure that these are real. And also discarding SNPs in places of the genome where there are potential issues such as collapsed repeats and so on, because then you start to get these kind of haplotypes collapsing on top of each other and SNPs that don't necessarily make any sense. So put a lot of effort into getting rid of that kind of thing.
All right, thank you. With dual hybs, is there a line effect? For example, do certain cultivars or land races of one species ruin calling for the mate in the pair?
I guess in principle that could happen, but it would probably be only affecting a few SNPs if that were the case. I think overall, again, as I said, the attrition that you see in dual hyb, when you're genotyping large numbers of samples, especially when you have related samples, even if you lose some SNPs because of interference like that, you can actually use imputation to get back those SNPs lost through attrition, as long as you've genotyped a large number with the single hybs as well to use as references.
All right, the next question is, in SNP chip genotyping data, how do you consider gap or null allele? For example, how do you proceed after identifying a particular SNP if we want to go to the details of the loci?
Yeah, I mean, okay, so null alleles, that's a really good question, because obviously in the SNP chip, it's hard potentially to differentiate between an actual null allele, i.e. that SNP is not present in that genome, and a miscall. So actually that's something that we've considered, and we want to be able to detect these kind of things because it's presence-absence variation, which is obviously there's quite a lot of. So that's partly why we build in that redundancy. And the redundancy means that because we have, say, 2 or 3 SNPs targeting a particular tag SNP set, if all 3 of them are nulls, we can actually sort of say with more confidence that that is actually a null allele. And so, this is something that we're just starting to process now.
Okay. So the next question is, how does the array work on integressions from wild species, Ventricosa, Penoperum, and others? I'm not sure I pronounced that correctly.
So, I mean, as I said, we started with, in wheat, we started with a very diverse data set, and we called SNPs. And the SNP selection algorithm that I described, which is based on the size of the tag SNP set, when you think about how that's actually working and what the first, the most informative SNPs that I described, where these are coming from, these are most likely, I believe, large integressions. So I think that integressions are probably the first thing that we tag. For example, SR36 in wheat, which is most of chromosome 2b, of course, this is tagged multiple times because it's so large. And I think that my feeling is that in integressions in particular, we actually tag very, very well.
Okay. Since linkage to disequilibrium is population dependent, what do you think is the drawback of utilizing this approach in diverse panels, such as land races, which were not part of the design to start with? So a continuation of the land race issue.
No, so actually land races were included. I mean, in this diverse data set that we started with in wheat, land races were part of that set, so it's certainly covered. And in barley, obviously the data set was comprised mostly of land races. So we do capture land race diversity. So I guess the question then is, how is it going to perform in material that is outside the diversity of what we're capturing? Okay, so in that case, I mean, we've made our best efforts to actually try to capture diversity as best we can. Now, of course, the LD capture is not the only thing happening. Its haplotypes are also important, and the LD, the SNP selection algorithm that I've described through LD, actually is a kind of nice way to sort of capture a set of SNPs that produce meaningful haplotypes. But actually, we can still see, for example, in exotic material, completely distinct haplotypes, right? And that's partly why we've included some of these SNPs from things like tauschii and the tetraploid wheats, because those SNPs being present, although in a highly sort of divergent line, we'd expect to see a distinct haplotype there anyway, we're actually including SNPs that are particularly enriched in that haplotype to sort of accentuate that distinction. So it's not just at the level of SNPs and tagging this and that SNP, but actually when you get very divergent haplotypes, it's actually very clear. Like for example, in barley, although we didn't use the wild accessions in that data set for the purposes of SNP selection, what we can see if we actually look at how those samples separate in a PCA, they're actually distinct. And so wild barley will separate in a PCA because those haplotypes are so distinct.
All right, great. Will the exome data for the reference panel be made available to the public for imputing new lines genotyped with this array?
So most of those samples were published alongside the Hay et al. Paper in 2019. And I think I note about 300 samples that are unpublished that we will be publishing alongside the upcoming publication describing this array.
All right. So is there a chance the multi-species pulse chip will be made available to the public?
So that's accessible to pulse researchers in Australia. I'm not completely sure about accessibility to the public generally. That's something that we'll have to answer offline.
All right. So there were a few other questions that came in that we were running out of time, so we won't have a chance to get to. But one last question that I have, does someone who wants to use Pretzel, is it freely available and anyone can use it?
Yeah. Yes, so Pretzel, okay, I'm glad someone asked this. It's actually open source software, so you can access it at GitHub at the URL that I'm showing here on the slide. So that's freely available. Anyone can download the source code. And the instance that we run at plantinformatics.io is also publicly accessible, and I invite you to go and sign up there, and we'll approve the account. And we're very keen to hear feedback from anyone who's interested in using it and your experience with that. We put a lot of effort into the development, and we continue to develop it.
All right, great. Well, thank you very much, Gabriel. And just as a reminder, anyone who did not get their question answered, there will be a chance to get a response offline, and we will post the Q&As on the YouTube, the IWGSC YouTube channel. With that, again, I want to thank you, Gabriel, for your time today, and appreciate everyone participating in our webinar, and we look forward to seeing you on our next webinar. So thank you very much and have a great day.
Thanks, Kelly. Thanks, everyone.